Hi, everyone. Thanks for joining today. My name is Jason Key at SB Grid at Harvard Medical School in Boston. So thanks for signing on. This is our kickoff to 2017 with our first webinar of the year. So we're going to do two sort of XFEL themed January webinars, both this week and next week. So be sure to sign on next week as well. I'd like to introduce Aaron Brewster. Aaron's going to be talking to us today about XFEL data processing with dials. Aaron is a project scientist at Lawrence Berkeley National Lab. Aaron, are you there?
Absolutely. Can you hear me?
Yep, I can hear you. So thanks for joining us today. Go right ahead.
Okay, excellent. Thanks to the organizers for this chance to present this information. I'm going to be talking about data processing with dials of serial crystallographic data. So dials is a diffraction integration program for advanced light sources, and it's a collaborative project between folks at Lawrence Berkeley National Lab in Nicholas Souter's group, including myself and Iris Young, and we're called Dials East. And then over at Diamond Light Source in the UK, we have our lovely collaborators at Dials East, David Waterman, James Parkhurst, Luis, Gwen Daff-Graham, and Richard. And it wouldn't exist without a great deal of work and intellectual contributions from other people, including people at XDS, Mosfilm, Andrew Leslie, and so forth. So greatly appreciated. So these groups have given other SB Grid talks. If you go to the SB Grid YouTube channel, you can see talks by Nick Souter and Graham on dials and cctbx.xfel. So today, what I'm going to do is I'm going to give an outline of serial crystallography in general, talking about some of the issues, challenges, and solutions we've been able to come up with for sample delivery and for processing still images. Then I'm going to talk briefly about processing data using dials itself, because Graham covered this in his talk. This is information about sweeps. And then I'm going to go over how to process stills. So serial crystallography can be thought of as a contrast between sweeps versus stills. So in classic protein crystallography, one takes a single crystal, usually cryofrozen, mounts it on a goniometer, and then rotates the crystal in the beam to collect a complete data set. But in serial crystallography, instead, what we do is we expose hundreds of crystals to upwards of millions of crystals in sequence. Each crystal gets exactly one shot, and we collect that diffraction pattern from that crystal. Usually there is no rotation involved when collecting these data because we're using a pulse source where you have a 40 femtosecond wide pulse of photons at an X-ray free electron laser, or at a synchrotron rastering through a chip filled with thousands of microcrystals on a microfocus beamline. And so because of this, we only get one shot per crystal. And why would we want to do serial crystallography? We've been able to use serial crystallography at XFELs and synchrotrons to do lots of interesting science. So X-ray free electron lasers deliver a huge amount of photons in a very small amount of time. And what that lets us do is study things like metal clusters, such as the photosystem 2 manganese cluster involved in the oxygen evolution cycle. The X-rays are delivered so fast that the crystal is destroyed before radiation damage can accrue. And so you get a diffraction pattern without any radiation damage. And it lets us study the oxidation state of these metals. We've also been able to use X-ray free electron lasers to study very, very small crystals to extend the resolution. We've been able to do time domain experiments, such as study the oxygen evolving cycle in photosystem 2 in plants. We've been able to do room temperature science, such as looking at the conformational changes of proteins. Sorry. Nope. And we've been able to study crystals in vivo. Much of this work can be done at synchrotrons as well, and so people have been investigating sample delivery mechanisms in such a way to do things like room temperature crystallography and time domain experiments. So sample delivery is probably one of the most challenging parts of serial crystallography. So I'm going to briefly explain just a few of the sample delivery methods that are currently in use. This one, for example, is one of the liquid jet injection systems where crystals are treated like a fire hose and thrown at the X-ray beam. In this case, a counter electrode for this nanoflow electrospinning device is drawing a solution, a crystal slurry out of a suspension into the beam using positive and negative electron differential. And so what we're able to do is run the X-ray free electron laser at 120 hertz and have enough crystal saturating that frequency to be able to collect data very rapidly, one shot per crystal. Other liquid jets exist, including gas nozzles, which focus an injection stream. Other groups have been, so that can consume a lot of samples. So other groups have been investigating ways to do things like use lipidic cubic phase to inject the sample in kind of a toothpaste-like system where very slow jet is extruded from a system and the crystals are suspended in a grease matrix. Other groups have used classical protein crystallographic approaches, doing raster scans. Here you can see the holes that the X-ray laser punched into this mesh, or doing helical scans. Other groups have investigated at synchrotrons, taking crystals and putting them onto mesh grids, and then either sealing them with membranes or humidifying them to prevent evaporation, and then taking this chip, mounting it on a goniometer, and rastering through, where you don't know where the crystals are, but you don't care because you just take small pictures of each one of these crystals and get the diffraction patterns, and hope that the crystals are randomly oriented on the mesh. We've had groups investigate microfluidic hydrodynamic traps, where you have these microfluidic arrays which trap individual crystals at known points, and then you raster through those. And finally, our most recent work is done using acoustic droplet injectors, where an acoustic transducer bounces crystals using sound waves onto a tape, and the tape translates through a pump region where it can be excited by other lasers or other devices before it enters an X-ray interaction region. And this lets us do time domain experiments. So all these different sample delivery methods have their pluses and minuses. They consume more or less sample, they have more or less path length. And so the idea is that you need to pick the sample delivery method that suits your system or your problem, your biological questions. And more delivery methods are being developed all the time and refined. So this is one of the biggest challenges of serial crystallography, is getting your sample into the beam. Once the sample is there and you've collected your diffraction patterns, then we come in with our group. Still images create lots of challenges for data reduction. I've kind of separated the two groups of challenges into physics-based challenges and technical challenges. So the physics-based problems arise when you're no longer rotating the crystal through the beam. You're collecting a single shot without rotation. So there are issues with spot prediction, partial reflections, and crystal-to-crystal variation. The technical challenges are things specific to serial crystallography. The data sets tend to be very large. We often have segmented detectors, and you can get exotic file formats that you need software to handle, such as XTC or Pickle, or complicated segmented CBF files. So I'm going to go over some of these challenges. Let's start with spot prediction. So spot prediction is what you need to do to be able to integrate weak signal. In order to integrate the weak signal, you have to know where weak reflections will appear on your detector image. So to do this, you use models. So the models involve a model for the crystal, including the crystal orientation matrix, a model for the unit cell, so unit cell dimensions and symmetry of the crystal. And then if you have that, then you also need a model of the detector, where the pixels are in space, and a model for the beam, including the wavelength and beam direction. And with all of these things together, these crystal rotations, these detector models, then for a given Miller index, a given HKL, you can predict which crystals will be illuminated on the detector. So spot prediction, two components, ray prediction, which depends on the quality of the models, and also index generation. In order to be able to predict where the signal is, we have to predict which Miller indices will actually diffract. And that depends on the beam character, such as that divergence, and the mosaic disorder of the crystal. So for stills, ray prediction, the quality of the models is actually quite difficult. For still images, we don't have two shots. We only get one shot, one image per crystal. So getting the crystal orientation matrix becomes difficult in a subtle way. The crystal orientation matrix consists of three angles of rotation around the X, Y, and Z axis of the crystal. The rotation around the Z axis is very easy to get. As the crystal rotates around the Z axis parallel to the beam, the entire diffraction patterns moves in concert around the beam. So refining the Z is trivial. But for X and Y, when X and Y are rotated, the spot locations actually don't change. Instead, X and Y change which reflections are in the diffracting condition. So I have an example here where we're looking at a perfect model. The red dots are where we're predicting spots to be. With a rot X misorientation, however, we change which reflections are being predicted. Some reflections shouldn't be predicted and some are being missed. We can compensate for this by increasing our mosaic estimates, which will cause more reflections to be in the diffracting condition, but we don't want to do that because then we'll be integrating noise. So instead, what we need is a stable refinement target to improve our X and Y rotation estimates. And so we add a new minimization target to our function. Let me explain what I mean by that. So as we refine the crystal orientation matrix for a diffraction image, we minimize the difference between the observed spot locations and the calculated spot locations. We change the crystal orientation until that difference is as small as possible. That's the conventional target, the squared disagreement between the observed and predicted locations. So for still images, since we only have one shot, we don't have that 3D information for locking in rot X and rot I that we would get if we had two shots. We have to add this other target. So to explain this target, let's look at the classic Ewald sphere diagram. So here we have the Ewald sphere. The points are the individual reciprocal lattice locations. This is the origin of reciprocal space. Ewald sphere is 1 over lambda in the radius. And this red reflection is exactly on the Ewald sphere, and so it'll diffract. So if you compute a ray from the center of the Ewald sphere to where that hits the detector, you'll get a reflection. But these other red reciprocal lattice points are close to the Ewald sphere, and depending on the mosaicity or beam divergence, they will actually diffract as well. So the rotational offset that you would need to do to rotate the crystals until these reflections are exactly on the Ewald sphere, we're going to call that angle delta psi. So we know that any observed reflection must be close to the Ewald sphere. And so we minimize, in addition to this positional target, we minimize delta psi for all the observed reflections. And this additional minimization target allows us to get a stable minimization, and we get a more accurate crystal orientation matrix. And therefore, we can predict the spots with more accuracy. Now, index generation, which spots to predict, depends on the mosaic disorder of the crystal. So in order to understand this, we need to look at how crystals actually behave in physical space. So let's start with the most abstract crystal. Imagine a perfect infinite crystal filling all of the known universe. That crystal, if you could get a beam to diffract it, would have its reciprocal lattice points would be just that, points, infinitesimally small. Real crystals have defined size, and they have mosaic blocks. And those mosaic blocks have average sizes, call it D effective. As the mosaic blocks get smaller, the reciprocal lattice points change in size, and they get bigger. Here we go. The mosaic blocks can also rotate relative to each other, which can increase the odds that they can intersect with the Ewald sphere and diffract. And the mosaic blocks can also vary in their unit cell sizes, and that can increase the reciprocal lattice point size. So these different effects can be resolution dependent or resolution independent, and they all convolve together to determine whether a reflection will actually interact with Ewald sphere and diffract. So let's look at a real example. So this is a diffraction pattern collected on a rheonics detector. And what I'm going to do is show the effect of decreasing the mosaic block size. So here we go. The mosaic block size is decreasing. And as we do so, the reciprocal lattice, we're able to predict more and more reflections, and they fill in. So we want to be able to determine what the mosaic block size, and we want to get it right. If we get it wrong, we'll either over-predict or under-predict the number of reflections. So how do we determine this mosaic block size or the mosaic parameters? For sweeps, it's really quite straightforward. Sweeps are awesome. We get all kinds of information from them. We can take a reflection, rotate it through the Ewald sphere, and slice it. And we can see as we rotate the crystal, the reflection appears and then disappears. And if we plot that as a function of the rotation angle, the integrated intensity, we see this rocking curve. To integrate the reflection, we just integrate the signal onto this curve, and that's our intensity for the reflection. But for stills, we lose all of that information. We only get one shot, which means we cannot estimate the mosaic parameters just from that one slice. In addition, because of this, every reflection from a still image is only partially recorded. We have to somehow figure out a way to inflate that measurement to the full equivalent. And so next week, Mona is going to be talking about how we do that. You can attend her talk through SP Grid. So how do we do this for stills? For stills, we take advantage of the fact that we can observe which reflections actually diffract. And we try to take that observation and determine the mosaic parameters that way. So to do this, we do what we call in our group here this trumpet plot. So here, what we're showing is in every one of these blue points is an indexed reflection. We're plotting the indexed reflections by their resolution, their 2 theta angle. And then on the y-axis, we're showing the rotational offset off of the Ewald sphere. At low resolution, reflections can be observed even if they're farther away from the Ewald sphere. But at high resolution, you don't see that as much. And this trumpet shape is observed time and time again. So when it's time to predict if a weak reflection will actually diffract, we can look and see where it fits within this envelope with reflections. If it's too far away from the Ewald sphere, then it won't diffract. So we treat mosaic block size and mosaic block rotation as counterbalancing parameters. And we combine both of those effects, and we find an envelope that will fit all of the reflections. The envelope consists of an effect due to the mosaic block size alone and an additional effect due to the mosaic block rotation. And there's a reference here to see a great deal more about this. It's actually, the trumpet plot is a very useful diagnostic tool. So here what we see is these are all the reflections from a diffraction pattern where the crystal orientation matrix is not quite right. And so because of that, you see a skew in the observed reflections and based on their calculated delta psi angles. And this skew is compensated for when we go and draw our mosaic envelope here, and which means we're going to overpredict the number of reflections that are on this image. So if we use after refinement, so for example, in this case, we allowed the detector distance to refine, we got a better crystal orientation matrix, and our mosaic envelope now tightly fits the observed reflections, which means we're going to be better able to predict which reflections are in the diffracting condition. So I'm going to skip talking about partial reflections because Mona's going to talk about that. So stills gives us problems with spot prediction, but also from crystal to crystal variation. When we measure hundreds of thousands of diffraction patterns, there's error in estimating the unit cell dimensions for each crystal. And that crystal to crystal variation is noticeable. So what we have here is a histogram of unit cell dimensions from 17,000 metalloprotein crystals collected at the LCLS beamline MFX. And for each one of the three unit cell axes, A, B, and C, what I'm doing is I'm histogramming all of the observed measurements of those axes, the lengths of those axes. Now, the width of these peaks is error in estimating that measurement, but what we also see is a bimodal population. So for this particular metalloprotein, there are probably two isoforms, two slightly different ways that the protein crystal can pack. When only looking at a one-dimensional histogram, it's hard to actually separate those two. But when we add a second dimension, and these are two-dimensional histograms where each pixel represents how many images were found at that combination of A and B axes, C or B axes, or A and C axes. What we see is it forms two clusters, and they separate nicely. And so for every image, we can assign it to either this cluster or this cluster based on if we refine the crystal orientation matrix, we can compute the root mean squared deviation of the observations minus the predictions. Whichever has the lowest, well, it's probably closer to that isoform. And that's a way that we can separate the images between their two isoforms. What we do from it at that point is still a matter of research. We haven't actually solved this problem completely. Oftentimes, we'll merge all these together and see what we can get. But we are interested in looking into solving each one of these structures separately and looking at the differences between them. It'll be very small. This is a very small change in unit cell dimension, but it's there. So non-isomorphism is a real problem and needs to be tracked. So now on to the technical challenges. Technical challenges when processing stills, I've covered quite in detail in this newsletter article in the computational crystallography newsletter, which can be found here. In general, we find that the data sets are very large. So you get these huge data sets that can be hundreds of terabytes. And so to be able to process them, we use big computing clusters, hundreds or thousands of computing cores. The rule of thumb is you need about 10 cores per hertz to achieve real-time data processing. And those 10 cores gives you a little bit of freedom. You can process the data a couple different ways. You can reprocess all the data at once. But if you have those 10 cores per hertz available, then you'll be able to keep up with the data processing during the experiment and be able to provide some kind of useful feedback. If you're going to process all the data after the experiment, then that's not so relevant. Additionally, we have a lot of segmented detectors, such as the CS-PAD detector at LCLS, which consists of 64 tiles. We have programs for refining those tile positions in cctbx.xfel and that use the dials refiners. And we also have software that interacts with these more exotic file formats. So if you have a file format and you want to know whether or not we can read it natively, then you just run the program dxtbx.printheader, and it will tell you what it thinks it understands about that image, and you can look at the information there and see if it matches up with what you expect reality to be. Okay, so with that, it's about time for the demo. Before I get into it, I want to recap something that Graham explained during his talk about the workflow for dials. So for dials, when you have a sweep, you can either process it using ZIA2, which will process the entire data set from start to finish and give you an MPZ file, or you can process it stepwise using all the same steps that ZIA2 would take if you use the dials option with ZIA2. These steps are as follows. So the first step is called dials.import. If you run that program on a set of CBF files, it will read all the image headers and try to understand the detector, the beam, and the orientation of the rotation axis of the goniometer, and so forth. And it will create for you a file called a datablock.json, which has all of these different models in it. You then run the program dials.findspots to find the strong Bragg reflections, and that produces a pickle file called the strong.pickle. The strong.pickle then has all of the bright reflections on all the images that it could find. So given a set of experimental models and a set of bright reflections, that's enough to index the data. Indexing the data will then give you a set of basis vectors, which describe the crystal orientation and unit cell dimensions. You can then refine those models. The crystal orientation is here in this experiment.jsons file. All of the indexed reflections with their Miller indices are in the index.pickle file, and then you can refine your answer and get a more accurate RMSD. After you have a refined set of models, you then integrate the data, and then you can export the result into an MTZ file and then scale and merge using pointless and aimless. So for stills, what we found is that before you can actually begin processing the data, you need to verify that the detector is properly calibrated. And we do that using an average. We take all 100,000 crystal images, diffraction images, and average them all together and look at what we get. I'll show you how to do that. And then we run the program dials.stills process. So dials.stills process wraps up all those other steps, kind of like ZIA2 does, importing, finding spots, indexing, refining, and integrating. Because each file is separate, multiprocessing is super important. We have some big computers here at LBL, and there's big clusters at LCLS. As you do more and more serial crystallography and the data sets get bigger and bigger, you kind of need computing resources to improve as well. And after we're done processing the data, we have two programs, cxi.merge and prime. Mona's going to talk about prime next week for scaling and merging the data together, taking into account partiality problems. So on to the demo. I'm hoping folks can still see what we're doing. Before I begin, I want to talk about the best way to get help with programs in the Dials toolkit. All right, so let's run dials.import. So -H, right? And here we get some nice help text and everything. And up here at the top, we see that you can run with the option -C, which says show config, which will show you the configuration parameters for this program. So if we do that, what we get, these are all the different parameters that you can run with dials.import. It's a lot of them. And just knowing the names doesn't help very much. So you can see up here at the top that it says that you can run it with at different attribute levels and different expert levels. So let's try and increase the number of attributes that we get for each parameter. And here we go. There's all the help parameters. There's all the help strings for each one of the possible parameters. And this is a great way to learn how the programs work and to figure out how to change how the programs operate as necessary. And if you want to even see more parameters, you can increase your expert level, just like Phoenix does, and there's just tons of parameters. So I'm not going to go into too much about what the parameters are. You can look at the documentation, or please be in contact with the developers to get help with the programs if you need it. So with all that said, the demo is going to be a data set that has been provided to us by Danny Axford at the Dynam Light Source. It's 11,000 diffraction patterns, or 11,000 images that were collected on a Pilota 6M using a chip technology that he's working on over there. In under 7 minutes, he was able to raster through all the 11,000 of these images on this chip. I've copied 10 of the images onto my laptop here, and there they are. They're CBF images that have been compressed. So we can run, as I said in the talk, print header on one of the files, and we can see how Dials is going to interpret the image headers for these data. So we can see we have a wavelength. There's a detector distance here for the panel for the detector. Here, 305. This is the origin of the detector, so X and Y and Z. And importantly, what we see is that these image headers include a goniometer and a scan and an oscillation. Now, I asked Danny about this. Still images don't have oscillations, and he told me that this is a problem with the image headers. And so when we go and actually process the data, we need to parameterize the program so that it ignores these oscillations because they weren't actually there. He says he's fixed it in subsequent data sets. So how do we get it? How can we let's actually look at the data. So we're going to use dials.imageViewer to look at the individual images. So it loads up. Here's the image viewer. And the first thing we see is a blank image. And so this is pretty much how serial crystallography works. You get a lot of blank images. When you're using a raster or a jet, a lot of the individual images have no data on them. So let's just page through until we find a diffraction pattern. And here we go. Change the brightness. And we can see the individual Bragg reflections here for this image. We're going to have to process this image using only the Bragg reflections you see, which isn't very many. Also from this image, we can look at the Bragg reflections. We notice they're pretty small. So if you click over here on threshold, well, it'll show you what the spot detection algorithm is actually going to see. So the spot detection algorithm, all these black pixels, these are going to be the actual spots that it finds. This spot right here is only 2 pixels in size. And it'll be important to make sure that we don't throw it out. So when we parameterize the spot finding program, we'll make sure that we're accepting reflections like this. If we go and we look at the reflection, the actual image, it's probably real. We don't want to get rid of it. We can page through and we can see more blanks, and we can see other images. So here's one. This one is probably multiple lattices. So 2 crystals that were exposed in the same shot. We can index images even if they have multiple lattices on them. Okay, so that's the image viewer there. So we want to get a sense of the entire data set. So to do that, we're going to average the data. So here's the command for averaging the data altogether. So dxtbx.imageAverage, path to the data set, verbose. And I'm going to create 3 images, an average, a standard deviation, and a maximum projection. I'll explain what those are here in a second. In addition, I'm going to use multiprocessing. I have 4 cores on my Mac. I'm going to use all 4 of them to be able to process these 10 images. Averaging the entire 11,000 image data set takes a little bit of time, more time than we have here for this talk. And so there's the 10 images. So instead of showing that result, I'm going to go, I've already averaged all the data. So it's like a cooking show. I've averaged all the data using 64 cores on a different machine. So let's load it up. So while that's loading, let me describe what these 3 files are. The average is, so for every image, we look at a single pixel and we average all of the pixel values that we see for that pixel in the entire data set. For the standard deviation, we do the same thing, but we compute the standard deviation. So here's the result of the image average. The image average tells us a lot about the data set as a whole. It tells us about the background, the average intensity of the background. We can see some artifacts. These artifacts here come from the silicon chip that Danny is using in his experiment. So we'll probably want to mask that out. We see other things that are happening here near the beam set, so near the beam center. These are bad pixels that we're going to want to mask out as well before we process the data. When we flip over to the maximum projection, so the maximum projection is probably the most useful image. Here what we see is the brightest pixel among all of the images for every pixel. And we get a virtual powder pattern from this result. And this virtual powder pattern is going to be dependent on the unit cell dimensions and the detector distance and wavelength. And given a set of unit cell dimensions and detector position and wavelength, we can predict which powder rings will actually exist. So if you go in here into actions and you click on show unit cell tool, it will start predicting rings. Now, the rings it's predicting right here aren't very useful. It's for a different crystal. So let's just quickly put in the actual unit cell dimensions for this protein. Let's change the highest limit that it's going to display. There we go. So 97.1, 90, and so forth. And this is in space group P6, which affects which rings are going to be displayed. And here we go. So based on this, we immediately can learn a few things about the image headers. We can see that the detector distance might be a little bit off because the rings aren't exactly hitting where they should be. So we can just increase the detector distance a bit if we want, and we can get a slightly better result. So typically what we do is we load these averages, and we can see the beam center and the unit cell dimensions and the detector distance, and we can try to get a sense as to how accurate our initial estimates are. We can try to adjust things to maybe get a better estimate before we start processing the data. So I'm going to close that and move on. Oh, there was actually one more thing I wanted to show. So for masking the data, it's important that we do that before we actually process the data. So I'll show you how to do that. I'm going to zoom in here on the part that needs to be masked. If you hold down the Shift key on the image viewer, you can draw little boxes. So let's mask it out like that. And then I'm going to mask out that little section. And you click right here on Save Mask, and it will write out a little mask.pickle file, which you can then use during processing. So we won't get, so that won't confuse the spot finding parameters. I also wanted to mention when I was talking about the size and shape of the spots, Art Lubimov from Stanford is going to be giving a talk with Mona next week to talk about IOTA, which is a program for trying to automatically optimize the spot finding parameters for an image. So I recommend tuning into that. So we're going to try to process these data. So what I've done is I've created a directory called results. And inside that directory, I've created a trial directory. I'm calling it trial 3. Why trial 3? Because I've already tried to process this data a few times. And each time I change the processing parameters, I use a different trial number. So the first time I processed it using some kind of default parameters. Then I added the mask. That was trial 1. Then I changed the spot finding parameters. That was trial 2. Then I changed the indexing parameters a bit. That was trial 3. And that's where we're going to start with today. So the program for doing the processing for each of the images is dials.stills process. And here it is. Dials.still process, path to the data, and then a fill file. And this is, if you use Phoenix, this is the same thing as the .eff file, the effective parameters file. I'm going to use all 4 of the processors that I have here on my back. I'm just going to run it. I'm going to explain what's in the process.fill file in a second. So it's doing a multi-processed spot finding, indexing, and model refinement. And there's a lot of output. You can look at it. A lot of information for each one individual image. But I want to go into this process.fill file, which is probably the most important part of this entire procedure. So fill files are organized hierarchically, where each block is separated by these curly braces. So let's start up here at Spot Finder. So here we see our min spot size filter that I mentioned earlier, 2 pixels. Let's keep all reflections that have 2 pixels in them. And then there's the mask that we made so that we can mask out the beam center. I want to mention that most of these parameters are documented in my newsletter article in more detail. So we don't have time for this talk to go through them all in detail. Just going to point out a couple highlights. During the indexing, in the indexing block, there's a known symmetry section. Here I've included the known symmetry for this protein. You don't have to. If you don't know what the cell dimensions and symmetry of your protein are, you can index without this section. You're probably not doing serial crystallography in that case. But if you don't know the unit cell, you can do this and then either cluster the results or histogram the results in a way that I'm going to show you in a minute. And then that'll give you an estimate of what most of the images, what their unit cell dimension is. You put that in here and you'll get way more indexed images because these are stills. You only get 1 shot per image. It's important to try to guide the indexer as much as you can. And so providing a known symmetry estimate really helps. Now, I also mentioned that these image headers have an oscillation in them, which is incorrect. But because of that oscillation, Dials thinks that it is a rotation. And so there are several points in here where I am just informing Dials that it's actually stills. So here on the indexer, here in the refinement section, and so forth. So I'm not going to go through it all in detail, but I am going to jump to the bottom of the file. So if you're on YouTube, you can pause it and then take a look at these parameters. And again, the newsletter article goes through these all in more detail. So the processing is finished in this other window. So here are the results. For every image, all 10 of those images, we get a data block.json, which has the image header information in it, the detector model, the beam model, and so forth, and strong.pickle, which contains the bright reflections that the spot finding program is able to find. If the image failed to index, that's all you get for those two images, for that image. So here, image number 32 did successfully index, and so you get index.pickle. You get refined experiments.json, which tells you about the crystal orientation matrix and indexing solution. It integrated the data. And these are all dials intermediate files. Down here, for image 32, it created this int file. This is a cctbx.xfel specific file that is used by our merging programs. So that's the direct input to prime or cxi.merge. We can actually use these to look at you can use dials.imageviewer to look at the indexed reflections and see if they're fitting. So I'm not going to show that today, but you can run dials.imageviewer and then index.pickle and datablock.json, and you can see which reflections were indexed. This is only 10 of the images, and as I said, there were 11,000 images in the full data set. So I'm going to tab over here. I'm logged into our computer here at LBL, which has 64 cores, and this is the processing result of all 11,000 images. It's just a ton of files, and it's too much to really be able to look through one at a time. And so what we've done is we've created some graphical tools to help understand what's happening. So I'm going to show the first one. So this program is called plot run stats from experiments. You give it a directory, in this case dot, you give it a few parameters like a minimum number of reflections for a hit and so forth, and you run it. And what it does is it will look at all of these intermediate files kind of one at a time and go into these intermediates, look at how many reflections were found that were strong, how many reflections were indexed, the I over sigma of each individual image, the diffraction quality, how far the image was able to diffract, and it will synthesize all of that into a plot that will give you a way to examine the hit finding success over time as the data is collected. It uses the file names to be able to sort the data over time. So in this case, they're sequential, which helps. So there's 11,000 of them. It's almost loaded them all. Here we go. And here's the plot. So this is adapted from our work at LCLS, so the standalone version here. So this top plot here, there's a point for every image. The x-axis is over time, and the y-axis is how many Bragg reflections are on that image. So this is a matplotlib function. You can just zoom in on a section. So here we see that the gray spots are images that didn't index, these gray points. That's an image that had over 500 reflections on it that didn't index. So why didn't it index? Well, you could go and try to find that image and take a look. So we have some tools that let you do that. This is still in development, but this is available now. So over time, you can see that as you rastered through this chip, that some regions of the chip had more images and some didn't. And then right here at the end, something happened, and the indexing rate went down a lot. So I'd be interested in looking at some of these images and figuring out what happened. This middle plot is a moving average of the indexing rate. You can see the indexing rate right here. It's pretty good, 60% indexing rate at some points. And then this plot down here is trying to look at the overall quality of each individual image. So for every image, there are 2 points on this plot, a red dot, which is the I over sigma at low resolution, and a yellow dot, which is the I over sigma at high resolution, in this case 2.5 angstroms. And you can see based on this, the average kind of signal that we're getting out of each image. And then the yellow line is something we're calling the high quality indexing rate. So if you indexed an image, did that image index to 2.5 angstroms? If so, we're going to count that as good, and we're going to plot what percentage of those images are good. And then there's some additional statistics down here. For example, how many images there are, how many of them could be considered hits. how many images we indexed, and how many of those went to high quality. Another plot that we provide is the unit cell histogram that I showed earlier. So this is called plot unit cell cloud from experiments. You give it a list of refined experiment .json files. Remember, this is the file that includes the crystal orientation matrix, the unit cell parameters, and so forth from a successful indexing experiment. And this histogram is similar to the one that I showed earlier, except for this data set, we don't see the bimodalism. I'll explain that in a second. Here we go. So here is the A, B, and C histograms that I showed before. You can see there's a slight left skew to this distribution. And I don't have time to go over this, but if you refine the detector distance slightly and doing an ensemble refinement, you can look in the newsletter article for more information about that. You can actually correct this skew and make this a symmetrical plot. So there's that. And then this plot is a three-dimensional cloud. Over X windows, it's a bit slow to operate, but you can, if there's clusters, you can see the different clusters appear very nicely in this cloud. Okay, so that's the demo there. As a final slide, what I want to do is show that we are currently implementing all of these programs into a graphical user interface. And we've deployed this interface at LCLS, and it's available for users to use. And it kind of wraps up all the different command line tools that I just showed you in an automated fashion. It will track new data as you process it, and it will submit those jobs to the processing queue for you. And then as those processing results will come in, it will do a live update of that run stats plot that I showed, or you can do things like this one I'm showing here, which is look at groups of data and look at whether or not they're isomorphous with each other. So what you do is you go in there and you can tag individual collections of data with different names, and some sort of like Instagram. So here what I've done is I've tagged a group of runs, a group of data as xtils2, which is one batch that came from one crystallization condition, or xtils4, which came from different crystallization condition, and or slightly different treatment. And you can see that xtils2 shows that bimodalism that I was showing earlier, but xtils4 here in green does not. So these are more isomorphous. So if I was, or other way around. So if I was doing the experiment, I would say, try to get more of these green crystals here. And then the sample preparation folks could try to drive the sample preparation towards that result. Okay, so with that, I'd like to finish. I'd like to acknowledge all the many, many people that have been working on serial crystallography. Specifically, I want to thank my boss, Nick Souter, and Iris Young in our group. I also want to mention Art Lubimov at Stanford, who has been working with Iris and I on these graphical interfaces, and all the folks at Dials who have been working on, especially James Parkhurst and Dave Waterman, who have been working on the refinement and the image header reading parts of all this, and Richard, who's been working on the indexing. And with that, I'd like to mention some of our funding. And also mention that we're hiring a software scientist. You can see jobs.lbl.gov to look into that. And we have a workshop here at Berkeley in February. So if you'd like to figure out a way to get over here and come get some hands-on practice, then that's available. Okay, and that's it. Thanks, everybody.
Great. Thank you, Aaron. For anybody who has questions, you can send those to me by chat. You can send them here in the chat window. Chat is in the bottom. It looks like a little bubble. I had one question sort of about, kind of more about the geometry of the experiment. You talked a little bit about processing stills with multiple lattices. Is the ideal number of lattices per image 1? Can you effectively process 2 in the same? I guess what I'm getting at is that the geometry would be that you're either collecting lots of blank images and then occasionally you get one single, or you could go to the other end where you're getting 2s and 3s and 4s, but you're getting diffraction on most images. Where would you say the sort of sweet spot there is?
Well, that's a great question. I would say that we don't really know where the sweet spot is. We do know that if you add too many lattices, you start to fail the index completely. Um, it's because for stills, for sweeps, Richard Gildea has a paper about this. He can pull out 6 or 7 lattices from a sweep of data because adding 3 dimensions to that means that you get way more information and you can use an approach to pull out all these different lattices. But for stills, it gets much harder. Um, I would say that 1 or 2 patterns is the ideal. And so optimizing the sample delivery mechanism so that you only get 1 or 2 crystals per shot, but you try to get as many shots with crystallization as you can, that's really, really hard. And
Yeah.
so that's what we strive to do. But if you have 2 or 3 lattices, we can handle it.
So you, in the plot that you just showed, you could conceivably filter out, more than 2 or 3 or 4 by tagging sets by number of lattices or something like that.
Well, the tags are more for groups of data, but we could do something similar to that. Absolutely. This software is so custom. Please be in touch if you want to do something like that.
So we have another question from Pete Meyer here in the group.
Hi, Eric. So when you had your slide up with clustering on the basis of unit cells, I'm wondering if you can distinguish clusters in unit cells from just having an angle on the crystal where you've got high uncertainty in the lengths of one of the cell edges.
Oh, that's such a great question. So, in fact, we have some very recent results about this specifically. What we've done is we've taken an entire group of data, and then we've sorted it based on whether or not an axis is parallel or perpendicular to the beam, right? And then we've histogrammed those individual groups separate from each other. And what we find is that if an axis is parallel to the beam, then that distribution of axes is very wide. So you're right, it is less reliable to get a unit cell axis that is parallel to the beam. And we can directly measure that. And then for perpendicular ones, it is more reliable. Okay, yes, thank you. And then what we've been able to do then is we've been able to do this, what's called this ensemble refinement. And an ensemble refinement means take one detector model and then 1,000 crystal models and refine everything all together in one big joint refinement job. And that requires a new and interesting way of handling a huge minimization problem. And then if you do that, this is new results that we just got last week. This problem is resolved. So the detector the detector position gets shifted slightly into the right position, and we're able to narrow the distribution of unit cells, even if the axis is parallel to the beam. So it's a really cool result, and we're really excited about it. And then we integrate.
Cool. And I hope I see the paper when it comes out.
Yeah. I want to mention also when we integrate using these new models, the I/O sigmas also go up quite a bit. So we're happy about that.
Excellent.
Great. We have another question from Duca Hextra, who is asking, when several stills are available from rotating fixed target, can you use the known relative orientation of the crystal between frames to better condition the estimation of the orientation, mosaicity, etc.?
Okay. Let's see if I understand the question. So you have, I'm going to give an example of when this was done, very similar to this. So in the here we go. So something like this. So this is a helical scan at an X-ray free electron laser. So each individual shot is a still image because it's a pulsed source. But because we know the rotation between them, then we could couple that information together. The answer is yes. Probably if I was going to do that today, then I would index each image separately and then do a joint ensemble refinement of everything together. But I think that there would be a way to parameterize dials in such a way that you could relate these using a rotation, but still use the mosaic and other still specific algorithms to index and integrate the data. So the answer is yes. If this is a situation you find yourself in, let me know. I can provide commands to do it.
Great. Any other questions? So, thanks. I think that was good. Okay, with that, it's 1 o'clock, so I think we'll wrap it up. Aaron, thank you very much for the great presentation, and I'm looking forward to next week. So everyone who tuned in, be sure to tune on next week. Aaron, thank you very much, and everyone have a good day.
All right, thank you.
Thanks.